where does it start and how does it take so long to get through, once you have a good idea, to turn that into an approved product? Well, it's the initial development of a new drug or formulation in the lab that takes, on the order of about a year, sometimes 2 years, once you know what the drug is and you try to formulate it and you test it then in a series of animal studies to determine whether this really has any hope, any promise. Does it look interesting? That can take a couple of years. And you go, well, that's not too bad. But then you really need to ensure that you have things in place so that it can be done safely, it can be done reproducibly, and it can be done in a manner where we can truly understand if it represents an advance for patients with specific types of cancers, in our case, blood cancers. So the first thing we do is we gather all the formulation data, all the animal study data that we think is exciting. That's where we have the dream. And we then go to the Health Canada and say, we want to test this in patients. And we've done some safety studies in animals, it looks safe. And then they'll determine whether that's sufficient to authorize it to go into humans. And now we can study it in people. That's just the first step. The next step is where we try to change that dream into hope. Hope that maybe we're actually doing something important to help patients with blood cancers. And so the first stage is a phase 1 trial, and that's where we want to make sure we can administer the drug safely to patients, and we find what the safety, what the adverse events, what kind of toxicities happen. Do you get upset stomach? Do you have headaches? Do you lose your hair? That's not a big deal for me. But we qualitatively and quantitatively assess the safety profile. And then we identify what dose can we administer where we think we're giving about as much as we possibly can without introducing toxicities that make your life really unbearable or that put you at a serious health risk. Once that's determined, then that's the dose that we recommend for moving into a phase 2 study where now we are treating patients with a specific type of blood cancer, in our case, AML. And in fact, not just with AML, but AML either in newly diagnosed patients or AML in first relapse patients. Those patients' populations all will respond differently to different types of therapies. So we try to really nail it down to a specific patient type. And we test the drug. We see how many patients go into remission. How many patients get serious toxicities? Is that a problem? Are the side effects as bad as the disease? Hopefully not. And we then look at how long are we making people live longer? And we'll do a couple of those studies to try and identify what patients do we think benefit the most from our treatment. At which stage, we then go into a phase 3 trial, and this is what's called a pivotal registration trial. We now have identified the patient population for which we think the study, our drug, will have the biggest benefit and make the largest improvement. And to show that, we need to compare it to what's called the standard of care. And for different types of leukemias and lymphomas and blood cancers, the standard of care will be different. So those standards of care are established in the medical community, and we have to do what's called a randomized trial, and this can be hundreds up to over 1,000 patients. So as you can imagine, those trials take quite a while, and even after registration, there may be additional phase 4 trials after the product is actually on the market, where we're trying to learn more about how it can be used more effectively. A key component of this, and we'll talk about it later when I describe our drug Vyxeos, or CPX-351, is randomization. So you want to ensure that you're not biasing the trial, and when you have a new treatment, that the physicians are going, oh, we want to give it a best chance, and we'll try and get the good patients into that arm, and because we really think it's a good drug. So what happens is we have a trial, it's randomized. We had our drug Vyxeos compared to the conventional treatment for AML, which is called 7-3. It's 7 days continuous infusion. You're hooked up to a pump, and you get this drug called cytarabine. And 3 days, days 1, 2, 3, you get this other drug called daunorubicin. It's an orange-colored drug. And you get a dose of that. So that's a 7-3 regimen. That's the standard of care for treating AML. And then we wanted to show that our drug was better. So when patients enroll into our phase 3 trial, it's randomized. That means they don't know what they're getting until after the patient has been enrolled. And then it's basically like the Lotto 649, there's a ping pong ball that comes up and it says Vyxeos or 7 and 3. It's a randomizer. So there's no chance for bias. And then we try to control every aspect about the clinical trials we can, so there is not the chance that a benefit could be associated with just a bias in the trial design.